Welcome once again to the AJP-Heart and Circulatory Physiology video table of contents for March 15th, 2015, or from the editor's desk. I'm highlighting 4 interesting articles today from this issue, so let's check it out. Michel's et al. Examined the role of vascular smooth muscle cell autophagy in calcium mobilization and contraction. Aortic segments from mice containing a smooth muscle cell-specific deletion of ATG7 were used to investigate voltage-gated calcium channels and potassium-mediated contractions. Voltage-gated calcium channels were more sensitive to depolarization, independent of membrane potential. Contractions with either potassium or a calcium channel agonist were significantly higher. Overall, the study indicates that defective autophagy in vascular smooth muscle cells leads to an imbalance between calcium release, influx, and calcium reuptake extrusion, resulting in high basal calcium concentration and significant effects on vascular reactivity. Erdos and colleagues tested the hypothesis that increased BDNF in the paraventricular nucleus elevates blood pressure and heart rate by altering angiotensin II signaling. PVN viral overexpression of BDNF increased blood pressure, heart rate, and sympathetic tone. BDNF also upregulated AT1 receptor mRNA and downregulated the MAS receptor message. These results suggest that BDNF overexpression in the PVN results in sympathoexcitation and hypertension by modulating angiotensin II signaling. Hoffman et al. Determined whether calcium entry via TRPM2 channels is required to maintain cellular bioenergetics. They used adenoviral constructs in wild-type TRPM2 loss of function, E960D TRPM2, mutant ventricles, and global TRPM2 knockouts. In slices from global knockouts, there was higher reactive oxygen species but lower oxygen consumption compared to wild-type. TRPM2, but not E960D, decreased ROS and restored oxygen consumption. Based on this and several other components of this study, the authors conclude that cardiac TRPM2-mediated calcium influx is necessary to maintain mitochondrial function and protect against hypoxia reoxygenation injury, and that calcium influx via cardiac TRPM2 confers protection against hypoxia reperfusion and ischemia reperfusion injury. Finally, Yancey et al. Addressed the question of whether the mitochondrial-targeted antioxidant mitoubiquinone, or MitoQ, reduced cardiomyocyte damage in left ventricular volume overload. A rat aortocaval fistula and acute myocyte stretch models were used. Cyclic stretch, or AV fistula myocytes, exhibited increased mitochondrial ROS production and decreased membrane potential that were significantly improved by MitoQ. Although there were marked improvements in mitochondrial structure and proteins, LV dilation and fractional shortening were unaffected by MitoQ treatment in AV fistula rats. The authors conclude that while MitoQ did not affect LV dilation or function in AV fistula rats, they suggest an interplay between cardiomyocyte mitochondrial-derived ROS and cytoskeletal disruption and connection to mitochondria. This was only a smattering of the great papers in this issue. Go to the journal and check out all the other studies published this week. Thanks for watching, and I'll see you in 2 weeks. Oh, and don't forget, send us your best work now.